Good afternoon, everybody. My name is Ben Jones. I'm the Director of Science at Alleyn School, and it's a huge pleasure to welcome you to thinking about one of these weekly SSLP lectures. Some of you may well have joined us for these talks during lockdown prior to the summer holiday. It's lovely to have a number of people on this afternoon. And it's a particular pleasure for me to welcome this afternoon Sir Jim Smith, who is a prominent and internationally recognized developmental biologist. I'm sorry about that. He's currently interim director of research and programs at Wellcome, visiting group leader at the Francis Crick. He's been director of science at Wellcome, director of Medical Research Council's National Institute for Medical Research, and director of research at the Francis Crick Institute. Jim is a fellow of the Royal Society, a fellow of the Academy of Medical Sciences, and a member of the Academia Europaea, and he was knighted in 2017. I love the fact that Jim in 2014 was named by London Evening Standard as one of 1,000 most influential Londoners, which is really quite special. I thought I was one of those people. But more than that, Jim is father to Gavin and Kirsty, who I personally had the pleasure of tutoring for the last couple of years at Alleyn School. Jim's wide-ranging research includes inductive interactions in vertebrate development and applying principles of developmental biology to stem cell differentiation. And we're very much looking forward, Jim, to hear aspects of this in your talk, how we came to be. Let me just mention the format before I finally hand over. Jim will speak now for about 25 minutes, following which Ed Marshall, a couple of our students, Ed Marshall and Lily Randall, will then chair questions. So why don't be thinking about the sorts of questions you could be asking Jim at that slot at the end. But thank you again for joining us, and thank you especially to Jim. Over to you.
Okay, thank you, Ben, for that introduction. Developmental biologist. If you can't say it, try embryologist. Means the same thing, starts with a different letter. So thanks very much for the opportunity to speak to you. I'm going to speak for 25 minutes, maybe a tad more if I'm not careful, but shout, Ben, if I rabid on for too long. I've called what I'm going to say how we came to be. And what do I mean by that? What I mean by that is to answer the question that every embryologist is interested in, which is the question of how a single cell, and what you see on the screen now is a single cell, that large orange-shaped thing is a single human egg surrounded by sperm, how that single cell divides and divides and divides, how cells become different, how some cells become muscle, some become bone, some become skin, some become brain, and so on, and eventually form human beings. And these two images here are from the mission, the mission that went into outer space, and this is meant to be human beings who are engraved on that spacecraft saying hello, in what is, I think, meant to be a universal gesture of peace. So the question is, how does that single cell on the left of my screen become human beings on the right? Now, this is a really complicated problem. It's also, in my view, a really interesting one. And I'm going to begin by illustrating the complexity by thinking about the numbers. So this is human development, the numbers. So that single cell becomes 40 trillion cells, 4 times 10 to the 13 cells in me and in most of you watching. That's quite a lot of cells. Amongst those cells, there are roughly 200 different cell types of the sort that I just enumerated to you, the cornea, the retina, fingernails, and so on and so on. So 200 different cell types. And the challenge is to ask how those different cell types form in the right place. How do you get the right number of cells, and how do you get them in the right place? To put it crudely, how come my left arm is the same length as my right arm? The left arm doesn't communicate with the right arm in the embryo in any way, as far as we know, but at the same length. The same happens in your legs. your legs are the same length. So how is that precision of development controlled? So that's the question we're interested in. Before I begin to answer that, there is just one mini fact that I'd like to emphasize, though, to emphasize the scale of the problem, and that is this. You'll know, I think, that one of the major cell types we have in our body is the red blood cell. And you may or may not know red blood cells are unusual because they have no nucleus, no DNA. So only 30% of those 40 trillion cells have a nucleus. Now, here's an interesting number that you can ask your mum and dad or your friends about. If you bear in mind that each cell that has a nucleus has about 2 meters of DNA in it, how long would the DNA in your body, in my body, stretch if you stretched it out in a long string? And if you were all there in front of me, I would get you to guess. I'd get some people to say, oh, from here to Putney Bridge, or from here to Cambridge, or to Edinburgh, or to the moon. Actually, if you stretch out all the DNA, all the DNA in your bodies will go to the sun and back 80 times. And one of the tasks that the human body has, therefore, is to keep an eye on that DNA, to make sure it doesn't become mutated, to make sure it doesn't cause cells to become cancerous. So that's one of the things that I think is worth remembering from what I'm going to say to you. We have a lot of DNA, and if you want to try and win a bet, I can promise you that it will stretch to the sun and back 80 times. So the challenge then is to say, these cells, how do we arrange them? And just as one more way of illustrating the problem, if each cell were a marble, then if you look on the left of the screen, as you can see it now, that's one of those big containers that you put on ships. So if it would take you 1,400,000 of those containers to contain all the cells that would make up a human being, and if you were to put those containers on the largest container ship there is, it would take 70 of those container ships to hold all those marbles, all those cells. So the challenge is, how do you get effectively those different colored marbles to form in the right place? And that's what I'm interested in. I claim, and it's true, that it's interesting in and of itself. It's a fascinating problem. And it's one that I've been interested in since I was 21 or something like that. And I'm now much older than that. Mr. Jones knows that I'm 65, so I'm much older than that. And it's a problem that I've been interested in for a long time. But it's important not only because of its inherent interest to me, but it's also important because 15% of couples have difficulty conceiving a child. 23% of human conceptions are lost in the first 5 months, and 2% of children are born with a major genetic defect. And it's only by understanding development that we can solve these particular problems. Mr. Jones mentioned my twins, Gavin and Kirsty, and they won't mind me telling you that they were born by IVF. And if you look at the middle image there on the bottom, that's Kirsty in utero. And if you look on the right-hand side, that's her twin brother, Gavin. So the kind of research that I've been doing is terribly important, not only because it's interesting, but also because it helps understand human development and therefore help people. So that's why we do it. Now, one way of trying to understand human development is, of course, to work on human embryos. And we can do this. We can get spare embryos left over after in vitro fertilization, and we can study them. And a colleague of mine here at the Crick, which is actually where I'm sitting at the moment, has been working on human embryos and has been using a technique called CRISPR-Cas9, which I'm not going to explain to you now, to study the role of a particular gene expressed in these early embryos called OCT4. And I've just got a couple of movies here to show you what I mean. So on the left, the one that I'm going to show you first is a normal embryo, and you'll watch that embryo in a moment divide into more and more cells, 4 cells now, 8 cells in a moment, and the embryo divides quite regularly, and you'll see in a moment that as that embryo divides, it creates a cavity in the middle, and you essentially have 2 kinds of cells. There's the cavity, and there are 2 kinds of cells. There are cells that are in the lower left at the moment. They become the embryo proper. They become the human being, and the cells around them will become the extra embryonic material, the so-called placenta. If, however, you knock out that gene, if you ablate the gene OCT4, you will see that that embryo makes a valiant effort to divide early on, but then things begin to poop out. So this gene OCT4 proves to be an important gene in the regulation of early development, and by understanding how OCT4 works, again, we might get clues as to why human embryos fail to develop normally. So one can work on human embryos. But of course, there are many things you cannot do with human embryos. There are significant, I'm trying to think of the right word. You know, when you're speaking, you forget words. Questions about the right, morality of doing it and so on. So there are certain experiments you cannot do on human embryos. So for that reason, quite a lot of the work we do uses embryos of other species, but we do that confident in the knowledge that the lessons we learn from those other species will also apply to humans. And here's a fantastic example. So at the top there, what you can see is an embryo of the fruit fly. And you can see that I've colored, or actually somebody else colored the head yellow and red, the thorax is purple, the abdomen is blue and then green. So from the front to the back, we go orange, brown, red, purple, and so on. And it turns out that there are genes expressed in the fruit fly along a chromosome, along a single chromosome, that control the expression of those different parts of the fruit fly embryo. That was discovered in the late 1980s. And it was 10 years later, because research can take time, that it was discovered that exactly the same principle is true in mouse embryos and human embryos. So there are genes that are expressed at one end of a chromosome that control the formation of the head, and there are genes expressed at the other end that control formation of the tail. And these genes, where I've got them colored the same, are homologous genes. They're very similar genes. So if there's a second lesson I want you to take away from what I say, it is that the mechanisms of embryonic development are highly conserved between species. And by working on one species, we can learn a huge amount about other species as well. And that's a really powerful thing that we developmental biologists or embryologists use in our work. And so one of the questions we have to ask ourselves when we work is, what species should we work on? And people work on all sorts of different species. Lower left, you can see the fruit fly, Drosophila. In middle, at the bottom, you can see a mouse. There's a worm on the right-hand side called Caenorhabditis elegans. Top left is a zebrafish. Middle is a chicken, and top right there is a frog. And different people work on different species because those different species offer you different advantages. They allow you to ask specific questions. And the work I do focuses on this frog. And this is the South African claw-toed frog, Xenopus levis. I think you can see why it's called claw-toed, because on its foot it has claws. If somebody ever says to you that it's a toad, T-O-A-D, they've misunderstood. It's T-O-E-D, South African claw, T-O-E-D frog. So that's what it is. And we work on this species because its embryos are terrifically accessible. They simply lay them in water. They lay many of them. They divide very rapidly. You can see them dividing there in front of you in complete synchrony. Because I understand these embryos well, I can tell you what different cells in those embryos will eventually become. I can mark them in one way or another, and I can tell you that those cells will become skin, those cells will become gut, those cells will become muscle, and so on. So the accessibility of the frog embryo, the fact that there are lots of them, the fact that I can map the different regions of the embryo and tell you what they will become, this makes the frog xenopus an enormously powerful system for those of us interested in early embryos. The question of how different cells in the embryo become what they become. So I'm going to tell you a story, one experiment about the development of the frog, and in particular, that question that I alluded to at the very beginning, which is cell number. How does the embryo control the number of cells that it makes? Because that's an important experiment. If the cells in my right arm divided more frequently than the cells in my left arm, then I would have one long arm and one short. And sometimes that happens, of course. Look at the fiddler crab. So there are examples where that happens, but in human beings, in vertebrates, the left and the right arm are the same length. So how am I doing for time? I'll speak for another 10 minutes, Ben. So here's a question to do with the early cell division cycle in the frog. And what I want you to do in a moment is look in that square on the left-hand side, in which I'm going to show you a dividing frog embryo. And what I want you to do when I get that embryo developing, as I will with a click, is to count the cell cycles with me. And I know I can't hear you, but I hope you can hear me. So I'm going to click, and then let's count the cell cycles. Here we go. Ah, didn't work. I'll go back one. The bane of the Zoom speaker's life. Here we go, I hope. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and now they become random. Now, if you're used to looking at this, it's easy to see. You aren't used to it, so you won't have seen what I've seen. So I'm going to take it back with a bit of luck and forward, and then just take it through you one more time. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and then they become random. And the message from this is that in the early division of the frog embryo, there are 12 very rapid cell cycles that are completely synchronous. And it's the same in every frog. And after 12, things happen. The cell cycles become asynchronous. Cells in the embryo begin to move. And those cells that are in the embryo begin to turn on the expression of different genes. So the things that make cells different from other cells, that make my skin cells different from my heart cells, from my lung cells, from my kidney cells, is that they turn on the expression of different genes. And that's what's happening in that early embryo at that point. So this transition, this time at which the cell cycle changes, is a really important time in the life of a frog. And a really important question is, what controls that 12? Why is it 12? Why isn't it 11? Why isn't it 13? And it was about 40 years ago that two scientists called Mark Kirschner and John Newport came up with the answer. And they came up with the answer using a really elegant approach. You know, to solve interesting questions in science, you don't necessarily need enormous pieces of kit. You don't need powerful microscopes. You don't need to be able to sequence DNA. You just need a good idea. And this is what they did. So this is this paper by Newport and Kirschner. The paper was published in 1982, which in the world of science is a lifetimes ago. I was alive then, of course, myself. And what they did was this. So they took an embryo, that's under A. So that's one of these frog embryos, and that's a drawing of an embryo in section. And they took a baby's hair. I know my hair isn't a baby's, but they took a baby's hair. And the reason for taking a baby's hair is that it's incredibly thin and flexible. And you can take a baby's hair and tie it around that egg and pull it tight. And that's what they've done in B. Then what happens is you can see that in B, the nucleus of that cell, the part of the embryo that contains the genetic information, is confined to one half. And that half embryo on the right-hand side divides and divides and divides. And eventually, those cells become small enough that, as you can see in D, one of the nuclei pops through into the other side, sort of like that. And then what you've got is you've got an embryo consisting of two halves. One half has got fewer cells than the other half. And what happens is that this mid-blastular transition, this point at which the cell cycle slows down, occurs first in the right-hand side as you look at it, and later in the left-hand side. And when Newport and Kirschner thought about this, what they realized was that the important thing that times the mid-blastular transition, that times the slowing down of the cell cycle, is the ratio of nucleus to cytoplasm. So I pause there to let that sink in. And the next slide gives you an illustration of what I mean. So this is, again, an image taken from their work. So imagine that in that thing that says 11 cleavages, you've got some substance in the nucleus. At that time, we didn't know what it was, but it's limiting. There's a certain amount of it. And in the nucleus also, denoted by those black bars, you have DNA. And as the embryo divides and more cells are made and more DNA is made, that substance binds to the DNA. So you see those balls binding to the DNA. And while the balls are in excess, while that mystery substance is in excess, the cell will divide rapidly. But when it's all sequestered by the DNA, when it runs out, then the cell cycle slows down. So this was a model put forward in 1982 by Newport and Kirschner, and things remained at that point for some 30-something years because we simply didn't know what that substance was. People looked, people didn't have a clue, people couldn't find it. Until, obviously this is my work, I wouldn't be talking about it with such a smug look on my face, until work by my colleagues Clara Collart and Phil Zegerman. And were I to look over there, you might see Clara walk past the door at any moment. What Clara and Phil did was reason to themselves that the material that might be running out might be something that is involved in the control of the cell cycle that makes cells divide. And they just did an experiment where they thought of every single protein known to humankind that might be involved in the regulation of the cell cycle. And there were 4 that they discovered at the bottom of this slide called replication initiation factors, with the unlikely names REC-Q4, Treslin, CUT5, and DRF1, whose levels remained the same or decreased throughout development. And what this is a thing called a Western blot, which depicts the levels of particular proteins at different times during development. So reading from the left to the right, you're advancing through development, and the intensity of the band you see of that little horizontal line gives you the level of the protein. And you can see that Treslin, REC-Q4, CUT5, and DRF1 decline, if anything, whereas the other proteins, if anything, are going up. So they reasoned that this REC-Q4, Treslin, CUT5, and DRF1 might be those limiting factors. And they resolved to find out whether that was the case. And the first experiment they did to show this is shown on the right-hand side. This is a picture taken from a real science paper, and it shows you that if you take an extract of cytoplasm from these cells and put into it different numbers of nuclei from cells and look at the replication activity, the ability of those nuclei to divide. When you don't have many nuclei, then there's enough of these factors and they will replicate. And that's what you see at 400. But as you get more nuclei, then that replication material runs out. And so the replication extent decreases. What they then did was to take these mystery proteins and add them back. And lo and behold, up went the level of replication, indicating, to my mind, very clearly that these are the proteins that are limited with respect to early replication. So I thought that was a terrific experiment, and I'm going to show you two more, and then I'm going to stop. So the next experiment's really cute. What you do is you take single cells from one of these embryos that is about to undergo that transition. And that's, I don't know if you can see me circulating the pointer on the screen. Ben says you can, so that's great. So we're taking single cells from an embryo that we'd previously injected with a precursor of DNA that we can detect, this thing called IDU. So we take these cells, and every time these cells synthesize DNA, they include in the newly replicated DNA some of this IDU that you can detect. And then what you do is you take those cells and simply stretch out the DNA on a microscope slide. And look, what you can see here in a control embryo is you can measure the extent of replication and the size of the gaps between the replication, the extent of the replication and a gap. And what we found is that in a control embryo, we had this much replication, but when we added the 4 factors, we had more replication. Again, showing that in the control, replication is limited. Add your factor, you get more. And lower left from here, the average track length is the same. So the speed of replication is the same. The gap between the labels is less, indicating that the frequency of initiation has increased. So what these factors do is allow the embryo to begin to replicate at more sites. And once replication has begun, the speed is the same. So my final slide is this one. And this is another movie, and it's simply to show you that we can make things work in the embryo proper. So here we've got 2 sets of embryos. One is a control, and the other is embryos where we've injected these 4 factors. The prediction would be that the control embryos would divide in that synchronous manner that I showed you, and then slow down. And when the embryos slow down, because the divisions halve the size of the cell every time, the cells will be a particular size. The prediction is that if we introduce these 4 factors, then there will be additional cell divisions, and the cells should be smaller. I'll just see if Mr. Jones gets that, if he's going to nod. He's smiling and he's nodding. Thank you, Mr. Jones. So let's run the video and see what happens. So you can see them dividing, and at the moment, I think the two rows are indistinguishable. But what you'll see as time proceeds is that the 3 embryos at the top, these ones, will go through an additional series of cell cycles compared to the ones at the bottom. And I think you can see that now at the end. These cells at the bottom are larger than these cells at the top, bottom top, because they've been through an extra cell cycle. So what we have done is we've worked out how it is that the embryo slows the rate of cell division at that particular stage. The answer is that it uses up its share of these 4 factors. And what we can do now, of course, is, having shown this in a frog, we can begin to ask, to what extent is this the case in other embryos, including, of course, other vertebrate embryos, and ultimately human beings. So what I'm going to do is stop there, because it's 4:33. I've probably rabbited on long enough, and I think it's probably more interesting for you to ask me things rather than for me to rabbit on. But thanks so much for listening, because I love talking about my work. You know, I enjoy my work. I'm excited by my work. So I hope you've got a little bit of the excitement of it. And I'd love to be able to show you where I work at the Francis Crick Institute, moving my screen. There's St Pancras Station over there, the British Library there. When you leave school, when you go to university, if you become a biologist, think about working in the Crick. You won't regret it. Thanks very much.
Thank you very much, Sir Jim. I think we've got some questions to ask you now.
So I've stopped sharing my screen now as well.
Perfect. Okay, so I was wondering, to what extent do you think that we are the product of our genes or our environment?
Oh gosh, that's a good question. To what extent are we the product of our genes or if I had identical twins rather than different twins, I would be able to answer that question much more easily. But I haven't. One's a boy, one's a girl, and they're very different. I mean, it's a bit pat and it's a bit of a cop-out, but of course it's both. Physically, I must say that physically, one of the things that always shocks me is the way in which human twins are as identical as they are. And if you understand a bit about human development, if you understand how you make a human face, for example, and all the cell movements that are involved in making a face and how complicated that is, to somebody who thinks about developmental biology, it's astonishing that twins look so similar. So we are a product of our genes physically. The extent to which we're products of our genes behaviorally and mentally is another question. And there I think, you know, although I said that the face is a complicated structure, so it is, but the brain is an even more complicated structure. So although physically, genes certainly determine who we are and what we look like, I think the variability in development and the way in which we can then be subject to the environment in which we live, I think that plays an important part too. So I'm sorry, Ed, it's both.
Thank you. We have another question from 16, who was wondering what the reasons for 23% of human conceptions being lost in the first 5 months were, if you knew about that.
Well, there are many reasons, of course, and you know, part of them are to do with the fact that some embryos may develop with an additional sperm, and that is not good for development. Some embryos may have derived from oocytes where the meiotic process didn't provide the egg to be fertilized with the right genes. Sometimes the mother may have experienced an adverse event of some description, but really, you know, the answer lies in the fact that it is an enormously complicated process with so much that can go wrong. And the thing that astonishes me is less that 23% of embryos, you know, don't make it. It's more that, you know, 77% of them will make it. And I think, to me, that is an astonishing fact.
So another question from Michael saying, does an additional cell division ever happen without intervention? What effect, if any, will an additional cell division have?
That's a really good question. Thanks, Michael. So the answer is, in all the time I've looked at embryos, I have never seen one with an extra division. But what we can do, and as you saw in the slide just now, is we can drive embryos to have an extracellular division. And we can answer the question, what happens? Ask the question, what happens? And I can tell you what happens is that the embryo develops at that point very poorly and dies quite quickly. Interestingly enough, that death is not a result of simply the fact that it has too much of these 4 proteins, CUT5, DRF1, Q4 and Tresden. It's not that. It is the fact that they've gone through one extra division, because you can rescue that death by slowing down cell division using another approach. So it's as if you're putting your foot on the accelerator with these 4 proteins. But if you put your foot on the brake using another process and get back to 12, then the embryo develops fine. So if you're a frog, if you divide 13 times, it's bad news. If you were to ask me why it's bad news, I would struggle to answer that question because we haven't followed it closely enough.
Thank you. And then another question from Esme. She was wondering what some examples of how this research could help in medicine were.
Well, I think in vitro fertilization is one, and certainly I know, and I suspect many of your friends at school have also come from in vitro fertilization. It's been an enormously valuable technique to help people who can't have children. But going on from this, I claim that so one other piece of work I did back in the olden days was to identify some of the signals that pass between cells in the early embryo that tell them what to become. So there are particular molecules called activins or fibroblast growth factor that can tell cells in the early embryo don't become skin, become muscle, or become blood, or become heart. And those proteins, interestingly enough, work in a concentration-dependent fashion. Now, you can use those proteins that we discovered, and you can apply those proteins to human embryonic stem cells, and you can drive those human embryonic stem cells down particular developmental pathways and make them become the tissue of choice. So one really interesting thing we've just done in our lab is to take human embryonic stem cells, treat them with a cocktail of these factors applied at different concentrations and at different times to make them become an almost pure population of left ventricle cardiomyocytes. So these are the muscle cells in the left ventricle of the heart. And these are the cells that if you have a heart attack, they're the cells that are damaged. So through this work and through a long process of stuff, we've been able to take the lessons we learned from the frog, apply them to human embryonic stem cells, and we hope use them in the future to be able to treat people who have had heart attacks.
Question from Toby Lober Joshua. How did you get into this type of research?
Toby, so that's a good question. And I think, so there's a principle behind this. So when I finished at school, in what we then called the second year 6, year 13, I was doing maths, physics, and chemistry. I'd never done any biology at all. And when I went to university, I took a course that allowed me to do those 3 subjects because I wasn't sure which I liked, but I also had to take one extra subject. And my director of studies at university persuaded me to try biology. I'd never done biology at all because in my school, I was in the fast stream as it was in those days. And if you're in the fast stream, you didn't do biology. So I did biology and I loved it. And I was taken down a path and eventually had lectures by somebody called Peter Lawrence. Peter Lawrence is a very famous fly developmental biologist. And the way he described the work, the way he described what was called positional information, led me to work with somebody called Lewis Volpert, who is the doyen and the inventor of positional information. And I just moved on from there. So it was in a way a series of lucky accidents, as life is often a series of lucky accidents. But I think the important thing, if I were to claim anything for myself, it would be to be open to change, be willing to try something new, not to define yourself too young. So I think that's important. I don't know whether Ben would agree, but I think it's really important to be open for as long as you can in your life to different influences, because that certainly paid off for me.
Thank you. Miss Count was asking, she says, we are currently teaching embryonic stem cells to year 11. At what stage in the cleavage cycle that you showed us do scientists extract the stem cells from?
So do you remember that video I showed of the human embryo? The one where, and I described the time at which the embryo had an inner mass of cells and a surrounding bit. So those inner cell mass cells are precisely the stage at which you would take those cells out of the embryo, put them in a Petri dish and get them growing. So it's that stage.
Lisa asks, when twins are developed, is it because an embryo is split, so there is double the same genes?
So identical twins form when an embryo splits. So you can have an embryo and the embryo can be fertilized in the normal way as a single cell, single sperm, single embryo. And then for some reason that we don't understand, that embryo can split into two. Embryos are really good at regulating their size. So the split embryos know now that they have to develop independently of each other, and they will each develop into a normal size embryo, and they will be identical twins. So that's how identical twins are formed. Non-identical twins, like my twins, they form from separate fertilization events in separate eggs. So there will be two eggs, each will be fertilized, and off they go and develop independently. You'll find that in vitro fertilization or people of advanced years who have twins have frequently had them through IVF, because the tendency, certainly when we had twins in IVF, was, as it were, to be on the safe side, to put in two embryos in the hope that one of them would make it. We were really lucky and they both made it.
Thank you. And then another question from Toby Lover Joshua.
Toby, you're into it.
Can an embryo be damaged during division? And if so, will it lead to physical illness?
So yes, embryos can be developed, so can be damaged through early development. And that damage can be of various sorts. It can be mechanical, so I could take one of my frog embryos and cut a little piece out of it, or it could be chemical, or it could be radiation, all sorts of things. Quite often, the embryo is able to, as we call it, regulate. It's able to correct that mistake in some way. The embryo knows that there's something gone wrong and can fix it. But at other times, especially if the damage occurs later on in development, when it's too late, then that mistake cannot be repaired. And you think about things like, thalidomide, for example, where the mistakes were not fixed and babies were born with, for example, short arms and legs. So the earlier the damage, in a way, the better, because there is the time to repair the mistake.
So Anna asks, what plans do you have for research in the future?
That's a really good question. So as I indicated, I'm getting on a bit, and I might have to hang up my pipette at some point. But if I were to carry on, the thing that I'm really interested in, that is quite understudied at the moment, and if I were to set off on a new tack, it will be limb regeneration. So this is a really interesting question. If you're a newt, and I were to cut your arm off, or here, I cut your arm off, at the cut surface, a structure called a blastema would form, and the limb would grow back. Humans can't do this. Very young frogs can do it, but really only newts can do it, urodele amphibians. And I'd love to know two things. One is, how does that blastema know that it's got to make those missing structures and not make a whole new newt? So it must know something about the plane. There must be some sort of proximo-distal polarity. And the other one is simply, how does it reconstruct those parts of the body that were missing? How does it know to do it? So that's what I'd work on, limb regeneration, in the hope bearing in mind that the principles will apply throughout all embryos, in the hope that it might be then applicable to humans as well.
So I've got another question. What are the limitations of not being able to kind of use embryos as you may like to, or human embryos, and having to only use other species?
Well, you know, I don't regard them as limitations, because to describe them as limitations even, to me, smacks of thinking to oneself, I'd really like to do those experiments on humans. And I think, you know, there are experiments I simply would never do. I mean, let's just make that absolutely clear. So I think, so that said, I think there is an awful lot that you can learn from other, you know, we call them model organisms. It's not really the right word, but you can learn a huge amount from work on those other species that I showed you. And to be honest, I think that working on those other animals will A, keep me and my colleagues busy for a very long time, and will be applicable to human embryos as well. So, you know, we won't need to do the experiments on human embryos. We will be able to infer what we need to know from working on the various model systems. So I'm optimistic that we'll learn a lot.
Thank you so much. I think, if I'm right, we're out of time. So thank you to everyone who asked some questions, and thank you so much to Jim. That was super interesting. Thank you so much.
Well, it was a great pleasure. And if I can just say, you know, I've got kids just left school. I know what a tough time it is for you at the moment, and I send you all my very best wishes for this very tough time. And I send it, you know, not only to you, but also to your teachers, who I know are working extraordinarily hard at this time. And all I can say is, you know, good luck, and may you stay forever young, as Bob Dylan once said. So thanks very much for listening to me.
And then one last thing to say, there's another one of these talks next week on Beethoven, so tune in if you're interested in that.
Not by me.
Thank you.
Okay. Thank you very much, everyone.